All right, so morning, we'll keep it, looks like it's really small today. Um, but we'll keep this focused. I think it was focused on colocalization today. So I have some demo data sets loaded. Um, we'll also keep it a little bit shorter. I know last time it was a lot of information. And we can always break it down into smaller chunks. That's fine. So let me go ahead and share my screen. And all right, so I have a demo data set here. I'm going to show it in 3D here. And there's also this auto adjust button for the histogram. So that kind of helps reduce it a little bit. Maybe we'll make the blue spots a little more obvious. So we have this 3D data set, and there are these cells here in red. And then they have these proteins or something, some puncta that's inside of them. So one of the something that a lot of customers will ask about is being able to what's called compartmentalization. So do cell by cell analyses and measurements. And so what that means is that, let's say I'm curious about the amount of puncta that's inside this object. We would create an association between a segmented object here and then the segmented blue spots. Let me turn this red channel off. Blue spots kind of in here. And then we can get a count just for this cell, just for this cell, just for this cell, just for this cell. And we can also combine them all to get an idea on what is in the entire image. And so the way that this kind of the pipeline that goes with this is that first we would do a segmentation, and then we would create that association afterwards. Okay. So turn on this red channel, and I'll go into our analysis pipeline. Make this smaller. And I'll create a new pipeline from scratch. One of these, I think this would also be compartmentalized cells or particles, would also be a good sample pipeline to start with. If you have any sort of cells that are similar like this, like any shaped cell that has some other channel, some other stain inside of it.
Does all the license have all the pipelines showing up on the screen?
The pipeline, so this is the pipelines are maintained just for each image stack. So I don't believe that I haven't used, let me put this button in. I have not used this data set before, so that's why there's no pipelines listed in here besides this one that I'm going to create right now. But all of the images that you do load will all have these sample pipelines that are ready for you to just preload. That's with any SIS file.
Okay, thanks.
That being said, if you create a pipeline for one image, and we can also do an export of the pipeline and then import that pipeline again, so this way you don't have to create it from scratch. Okay. But in this case, we'll create it from scratch. Save my image here quick. And as mentioned before, we for every step of this pipeline, we'll have to do an import ROI. In this case, let's see how big this file is. Oh, it's really small. It's only 2 megabytes. So we'll go and do the entire 3D image set here. Keep both the channels, and then because it's so small, we don't have to scale this down at all. So we'll do this, input ROI here. Gonna hit add operation, and then, well, I guess we'll kind of talk through kind of my steps that I'm going to walk this through first. So the first thing I'm going to do is get this outline of the red channel. So even though these aren't, maybe if I go in 3D here, even though that these aren't, doesn't look like it's a membrane stain. It looks like maybe there's some other cells that are tagged here. Point being that there's some gaps here that we kind of want to fill up and just get the border of this object. So we can take advantage of this really bright stain. You can see that it's kind of maxed out of this histogram. There's some information that's in the background, a lot of the upper limit of this histogram in this channel too, but it actually works in our benefit here. I don't think we have to do any pre-processing. So if we kind of think of the steps that we have to walk through, I don't think we have to do any pre-processing because this background around here, number one, is pretty noiseless. Like I'm not seeing anything here, and we have our histogram maxed out. We don't have to do any background correction, normalization. I don't think we have to do any of this. So we're going to skip this image processing step, and we can go right to segmentation. So with segmentation, there are a few options that we have. The first kind of, I always think of segmentation as kind of a, like go in ranks, and start with the easiest stuff first as like a first pass, or think about at least. And then you kind of work your way up to the more involved methods of segmentation that require maybe some additional input. So the most basic thing is this intensity threshold segmenter. And this is really just taking one feature, which is intensity, and you're segmenting based on that. So I'll go ahead and put my eyeball up here. So for something like this, and then we have our 3D viewer here. So we can kind of drag this depending on where we want it to go. This 4D viewer, this preview is newer. So that's why it just says that it's experimental. Again, when I apply this, it may not necessarily be the most accurate. Well, maybe if I do accuracy here. This may or may not be the most accurate segmentation result. So it's saying that we'll get everything, but we're missing some spots here. May or may not be the case. But we can just test it really quick. So we have our 4 objects, and you can see that it is, for this case, it was pretty accurate. It's kind of just going inside the border. So maybe we'll lower this a little bit. I'll put this eyeball back on. So the person to the group. And that looks a little bit more saturated here. So it looks like now I'm looking to make sure that it's kind of filling in some of these gaps. And the reason being is that if I do kind of reduce this, and I want to do a colocalization based on some parameters, like let's say I only want the objects that are 100% inside of this red object. So only these blue puncta that are 100% inside this red. And if we segment something like this, you can see it's not going to be 100% colocalized. So we had it about 60 here. Right. So maybe something like that. And I'll go and run it. Maybe we can, to make it look a little nicer, we can change the color of these objects. So right now it's set to just changing it to a cyan color. We can either change it up here in this color wheel, or we can just change it in the operator itself. So just to show you guys both ways, I'll hit this, change this color, and then we'll hit use random color. So that means every object that's created within this operator is going to have its own color with it. And we'll see this color wheel again pop down here. And then we'll change this to something more identifiable. So we can change the actual operator itself. So segmenting red cells. And in this case, or maybe we'll say segmented red cells. Okay. And we will run this. Now, each one of our cells are going to have their own color. It looks like it's kind of, maybe if I turn off this blue channel, looks like I don't see any red, maybe the faintest outline, but that's probably a good spot because we don't want it to be super oversaturated. Right. And then you'll also notice that this object table popped up a couple times. So this is going to usually, well, I guess it'll always pop up in cases of where you run an operator. And this is interactive with our viewer here. So let me zoom out here. If I click on any of these, it'll bring me to where that object is in the table. I already have some features selected here. So I have the sphericity of this, the volume of this object. This is not a neuron, so we can get rid of that. And then down here, we only have those two features selected. So be fine with those two. Maybe we can also get something like maybe the intensities, maybe the mean intensity of this, and maybe the sum too would be useful. So all of these features are going to be automatically calculated. We'll just kind of select which ones that we want. So we see the mean intensity for all of these, each of these objects, I should say. And then we have the sum intensity for each of these. And then this also works both ways. So I can go and click on this and it will pop up now in the objects table. I'm working on a small screen, so usually I'll have this up at the same time, but small screen. Right. I'll kind of go through some of the other ways that we can segment it, just because there are a few ways that we can get objects segmented. This is just the most easy way. Well, maybe I'll keep this running and then we can kind of compare the results. So if I go to add operator, another method that we can use is maybe we can try this blob finder. So it says that these blob finder detects small roundish objects in dense and noisy environments. So we can see, we'll definitely want to use that blob finder to get these blue objects out because they look pretty blobbish, right? They're small. They have some sort of spherical structure to them. Maybe not perfect, but close enough. But I'm wondering if we can also kind of use that to get these larger objects because they also do look like large blobs. So we'll go here. We'll just try it out. Go into our 2D viewer, and then our measure tool will pop up. I can also select the measure tool here. Right. Then there is an upper limit to this. We're not quite at it. 16 microns is the limit. So in this case, it might actually work. So let's see. Like this eyeball as a preview. You can see it's getting these three fine, but you can see that it's messing up here. It's splitting a little bit too much. So there are some other, oh, maybe I'll also hit this accuracy up here too. Okay, maybe I'll go down here just to kind of show it. So for here, you can also see that we have some other inputs. We have this probability threshold, and then we have this split sensitivity. Not sure if we went over this last time, but I'll go through it again just really quick. We can hit this eyedropper, kind of use it to drag across an object of interest. So I'm just clicking here, holding down, letting go here. You can see that's kind of picking up the range. So if I kind of drag it over some of these darker areas, you can see it's picking up that full range. So thing like this, right? And then we also, right now it's showing a layer of probability map. Let's go back to result. And you can see that the borders of these are fine, but there's still this splitting issue. So let's lower this value to maybe 50%. And you can see it kind of fixes it up a little bit. Now we can go to accuracy. We will run it in a second. The other thing I want to point out with this blob finder is there is some normalization. And this is going to be, it's going to normalize based on the histogram. And it's kind of asking you, where do you want to take that histogram information from to normalize on? So in this case, it's saying take it from the first time point. But in our case, we don't have any time series data. If we did, you would see another one of these drag bars. These sliders would be just underneath it. And this is our 3D slider, our Z slider. So in this case, first time point is going to be the same as per time point because we only have one time point. And then manual would be picking that range that you want to set yourself rather than normalizing automatically to a certain frame or a certain time point. So we'll just say first time point, just because it's only one time point here. We will call this segmented via blob finder. We'll change this color to use random again. And let's see if we get a similar. Okay, so we're still getting 4 objects. Now we can kind of compare them to each other here just to see what looks a little bit better. So let's figure out which object is which. This one here. Okay, so these two are the same, and you can kind of make out that if I look at the bounding box, and you can even see the overlay of some images, you can see that this segmented red cells using an intensity threshold is a little bit wider than using this blob finder. You can see that it's just a little bit, this blob finder is a little bit on the inside, and you'll have to go through and kind of check and see which one is more representative. And it looks like maybe this intensity threshold is a little bit better because it's kind of filling in these other regions versus blob finder's kind of smoothing this out a little bit. But if we look at the values here, a little bit different, at least in terms of the volume. Mean intensity and some intensity isn't that much different, but it is something to keep in mind that there is going to be a little bit of variability depending on how you choose to segment it. Another thing too is this blob finder is and the intensity threshold segmenter are both using some sort of histogram or some value. So this intensity threshold is using a single value versus this blob finder is using a range of values here. So you'll have to kind of make the decision on what looks best for your data or what makes more sense. Another thing that we can do is, I think I mentioned this last time, but we have this Cellpose based segmenter. Same exact idea as the rest of them. I'll do it just for demonstration purposes. Get rid of this minimal area. I'm just going to go through this. Use a random color here. All this segmented via Cellpose. Okay, kind of more or less the same. And Cellpose, again, this time Cellpose, the difference between Cellpose and these two other operators is that it is it's more sensitive to intensity changes. So whereas with these two operators up here, we kind of have to set the histogram or set a value. Cellpose is more resilient to these changes because it is a pre-trained deep learning AI model. So if you have some objects that are really low in intensity, some objects that are really high in intensity, for example, if you're doing immuno, sometimes that immuno is a little bit variable depending on the trial that you're, if you do it one day, the next day may be a little bit different. This kind of avoids some of that.
What are the other choice in the Cellpose model, the menu?
Yep. So we have a few different models that are pre-trained. I'll go into that actually right now.
No, I mean, now in the screen they shows the CP. So if you click on that, what are?
Yep. So there's a few different models that are built into Cellpose, and I can actually go to this help menu and I'll show that's not this one. So if I'm just hitting F1 with my cursor held over this operator, and that brings me to this help system here. Within here, we have some publications that you can reference if you do use the Cellpose operator. But down here, it says that it has a few pre-trained Cellpose model. And then these are the 5 different ones that you saw. You see here, so CPX, Cyto, Cyto2 nuclei, and then the CP. And then we can see kind of how they were trained. So the CP and CPX was trained on fluorescent cell images. CPX was trained on a wide variety, and I think CPX was trained on also just some regular images like a bowl of fruit or something like that. Cyto and Cyto2, it says that they were trained on 1 or 2 channels. So if they had a nuclear channel, maybe you would want to use a Cyto2 pre-trained model that's been trained on 2 channels. Or if you just have a single channel, maybe the Cyto would be better. And then nuclei was trained on 2 channel images. First channel is to segment, and then it just gives you some more information about these models. Okay, so these are useful. This is definitely useful information to know how these models were trained. Usually I work with whatever gives you the best result, it gives you the best result. So even if you're looking for nuclei, let's say you have an image of just nuclei, and if you run this CP model on just that nuclei channel and it gives you the best result, then use that one. It may not make sense to then use that nuclei if the result's a little bit worse. And we may only have just one channel image with this too. Okay. But it'll give you some information on this. And then there's also this other option, this external model down here. And this would be where if you did want to train your own custom Cellpose model, you would essentially start with one of these. And let's say that the result is, I'll just make this really bad just to show you. 5, eyeball up. And then, so let's pretend we had a model like this where it just wasn't really getting the result that we wanted, no matter how many of these operators we change. There's also some more parameters that we can change down here. What the custom Cellpose model, so this external model, would allow us to do is we would have a GUI that looks just like this. It's opened in Anaconda, but the image in the GUI would look, you'd see the same image. We would see our predicted objects that if we were to segment it, this is kind of the result that we would get. And then all we would do is go in and say, this is actually how that should be segmented. You'll then kind of correct it, and then it'll retrain automatically once you place the correction in, and then these three would then go to whatever you manually adjusted it, and then it would retrain with that in mind. So that's kind of how you build a custom Cellpose model. But it's always best, I think, to kind of start with these parameters the best they can be. Here we have a result here. Put these objects back on. You can see them in 3D again. These look a little bit smoother because, again, it's a little bit more resilient to intensity changes. Make this see-through. Turn off this blue channel. Whereas if we were to do something like this, what was that first one? This intensity down here. Let's change these to a fixed value just so we can see them. We'll make these solid. Kind of go over here, you can kind of make out the difference. So Cellpose goes a little bit above it. Maybe we can refine that value to be like, I had it at 15, which is probably a little bit too high. So maybe I would make it like 10. Show you that. Here. Use this. Cellpose to be glass. Kind of more or less the same. But again, kind of think about your question. Think about if you need these like grooves and these ridges to be pretty precise, then maybe you would want to go with an intensity threshold or go with a blob finder. That's going to take into consideration the intensity a little bit more than Cellpose would. Okay. So I think for all of these, we'll stick with this this first operator, this intensity threshold operator, and we'll get rid of the blob finder, we'll get rid of the Cellpose. And then, so that's kind of our first step that we have now. If I run this again really quick, we now have our 4 objects, and I want them to be pretty precise on the intensity that we have coming from our channel. Okay, so the next thing would be to get now these second objects, these blue objects here, and I kind of hinted at it before, but we're probably going to use Blob Finder for this because they're meeting the criteria. They are these small roundish objects. So we'll find the channel. And I'll go into 2D to get an idea on how big these blobs are. I'm going to look maybe something like this. Here. And even you can see it also has, it's a little bit smart too. The blobs are also able to split these objects that are really closely positioned next to each other. So you can kind of make out this is sort of one blob right here, and there's another tiny one just above it. And you can see that they're splitting. If you can adjust this to make it really split, like a lot, really heavily split, probably not what we want. Or we can make it really restrictive. Maybe we'll do something like 51%, I think is a good starting point. The important thing is, and maybe if I wanted to split these, maybe we can look in. Looks like we probably would keep these together. Okay. Maybe something like that is good. 50%. I have to reduce this to because there's another object there. Okay. Right. So we'll run this. Again, this we only have one time point, so we're going to keep it. These are both going to be the same. In this case, these are going to be blue blobs. And then we'll change these to be a random color. This way. Right. So we'll take a look at the result, but in this image, we have about 265 blobs. I'll go and hit these objects back on again, turn off the red channel, and we'll take a look on how it's doing. So maybe it's better to look in 3D too. We have our objects here. We can turn these to be transparent. Maybe we'll make them red because that'll contrast with the blue. It's called blue blobs. Next surface. Spread. Okay, now we'll make this glass out. That's why 100%. Okay. I'm just gonna look and see if it's kind of grabbing everything. I can make out the borders of these objects, but even if I can't, they can just select on them by clicking on it. And that looks like it's grabbing everything. Okay. So this is pretty close. You can always adjust this. Again, if Blob Finder wasn't really cutting it, we can use some of the other options too, like Cellpose or like a machine learning segmenter or something like that. But in this case, I think the Blob Finder actually works really well for a lot of these cases.
Can you show the results with just the surface rendering with a random color?
Yep. Go back in here. We'll hit random. Apply. I have it now on glass, but we can make this a non-glass shape to kind of bring them out a little bit too.
Yeah, thanks.
Yep. And we can do some filtering, either in adjustments of the values, but there are some really tiny blobs that are getting included. Maybe we want them, maybe we don't, but well, actually, I don't think we would need to because we're going to do colocalization. And if we're looking at colocalization, this is going to not be co-localized with it, right? So it'll kind of fall out anyways. Okay. So we have our object segmented, and now we want to create an association between these. So we'll go in here. We're done with our segmentation step. We have all of our objects segmented, and now we're going to do this use object step. And we have colocalization, but I think, we also have ways that we can calculate distances between objects. So not quite colocalization, but it's still pretty useful, especially if you're looking for maybe the location of where proteins are within a cell, or how far away is it from the nucleus. If you're looking at maybe like transport assays or something like that. You can also look at how far away is maybe you have like a treatment in a dish and you have a drug on one side, control on the other, and you're doing like neurons growing out. And maybe you want to see like from where you put the drug to where it's growing, maybe the distance between that. Um, so that would be in this distances here. And you would essentially pick your source, pick your target. In this case, it's not really a great example, but I'll do it anyway. So we'll say our source is going to be the larger object, and our target's going to be these blue blobs here. This could be useful after we do compartments. We can say if these are going to be a parent-sibling relationship, but it's not going to, that just kind of is going to maintain the relationships. But in this case, we're just going to get distances. And then we can choose how we want to save these. So we'll call this distance min, and maybe it'll be like example, I'll call it example 2. Here. Okay, so we have our distances. Then we can kind of break this down into further pieces. So if I go into feature columns, it's now going to create a new column here called what I just titled it within this distances. So it's called this example here. We can find then the information for each of these relationships between these. Okay. We can also break these down into groups. So let's say you wanted to bin this data, or let's say you wanted to, I can't think of an example right now, but if you wanted to bin the data, let's say any object that's 0 to 5 microns away, put in one bin. Any object that's 5 microns to 10 microns, put that in another bin. And any object that's 10 microns and higher, put that into a third bin. So one way that we can do that is doing group objects by feature. And we can do, we'll take the input with, in this case, it's distances. In this case, we'll say any object, and then we'll pick the feature that we want to adjust it by. Again, any of these features that we have in that table can be used, but we can also use this distances, for example. So maybe I'll say like the minimum distance between an object. You can hit create groups, select how many bins you want it to have, and then kind of the range with this. So if I kind of want to bin it into 3 different groups between 0 and 20 microns, so your first bin would be 0 to 5. Your second bin would be 5 to 10 microns, and then this would be 10 to 20. And run this. And there are 3 groups here. It'll then create kind of these associations with each other. And then we would have this group down here. So we'll have the parent IDs. You can also get the children ID. So any of the blobs that are within it. We'll also have, let's see, I gotta go down here. Parent IDs, children IDs, number of children. Well, these wouldn't have any children, would they? These would have the children. And then you'd have the ID of those objects within there too. Same IDs as here. So this is useful if you're going to export it as like an Excel file. You would be able to see just these columns, but you would still have the IDs related to one another. So then you can do your sorting after the fact there, even though you can't see the objects themselves in an Excel table.
What are the children?
Children are going to be anything that's it's just a group. So because we're saying take this large object and then find any of the blobs that are within it. It's not a great example because there's a ton of blobs within here. So it's going to say that pretty much this is going to fall into multiple groups. Because then if I select this.
When we group it, we group on the blue channel, right? The blobs.
Mhm.
So that
Yep.
group 1 should be all the blue blobs with the value from 0 to 5. Is that correct?
Yeah, the issue is that it's not a great example. Maybe for next time I can find a better example. But the issue here is that because they're inside of this object, it's going to automatically define them in that 0 to 5 group. So that's why we don't really have anything in this group 2 or group 3, because all of if we're starting with this object here and we're saying find it to the blue blobs, it's going to go by the centers, and anything within the center, it's going to hit all of these. And if I go and do a measurement, we only went up to 20, and the diameter of this is like 15. So it's going to kind of capture everything first. So it's going to say like these objects are going to be all falling within this 0 to 5 range because they're all contained within the object. This distances is really nice for cases of where you want to find like this object with respect to a further object away where they're not kind of contained within another. So I can find another example for next week of where objects that are kind of close together. Let me write that down really quick. I think next week is just going over movie making. So we can kind of like create that example really quick with distances. Example set, and then we'll create a movie on that afterwards. The reason I bring it up is because there's also within our colocalization function, there's also a distance operator. So I'll go back here. And the way that we do colocalization is through this compartments tab or this compartments operator. So I'll hit this. And this is a lot more simple in terms of its application. So I'll load in my objects of interest. And for here, this compartment is going to be just creating these associations between a parent object, which is going to be the larger object, and then the siblings, which are going to be these smaller objects. So I can take this red cell as my parent, which is going to be the first tab up here. And then we pick the objects that we want to co-localize with it. So I'm going to say these blue blobs. There's going to be then this option right here, which is going to be configuring how we want this co-localization to work. So in this case, we can say it needs to be 100% inside or intersecting at some point. I'm going to say we're going to do maybe 50% intersecting. And the reason being is that because there are some objects like this one up here, you can see it's touching, but in some planes in 3D, it may not touch. So in this case, it doesn't look like it's touching, but it probably is touching on another plane, like within here. So I want to make sure that we're still capturing it, even though that it is not fully inside of it at all Z-planes. So maybe we'll do it like 49. That being said, if we want to kind of capture objects that are nearby and include it into that association, like maybe we want to say we want this to be associated with this object too, we can then add in this close proximity where it's saying that any object that is not inside or intersecting in any way, if it's close by some amount, in this case it's 2 microns, it'll also make that association. So I'll run this here. And then we have this new tab called compartments. And we have now You can see our siblings are going to be listed here first. We have the same number of blue blobs here, siblings, -2, because there were 2 that were not touching these objects. But then in master detail, we now will have our parents up top and our children down bottom. So if I click on this, I find which one it is. So it's this one here. This is going to be our parent, and our children are going to be, and then you can see the parent ID is going to be 4. This is segment 4. We can see all of our children IDs. So there's going to be a ton here, but it's going to have 70 IDs associated with this. And these are all of the individual blobs that are part of that object. And then we can click on them further. See, these smaller ones are now being selected. The more that I select down here. Okay. So if I want just information on just this object with just its blobs that are inside of it, we can export or just pull information just about this specific object here. Same thing with this here. There are 68 blobs that are within this object here. And this one up here, there are 73. And then this one over here probably is going to have the least, 52, probably because it's cutting off some of these on the border. Okay. And then if we wanted to do like masking with this colocalization, this would be a second step. So we can there's a function called object math. And this is where it's going to combine object shapes. So this is kind of if you have like 2 overlapping objects and you want to find like maybe a colocalized region, maybe the volume of their overlap, this is how we would do it. So we would go here, we pick object math, and then you kind of think about what you want to do with it. So in this case, we're going to do colocalization. So maybe we would say just look for the intersections between these. And pick our object A, which is going to be this segmented red cells. We'll do these, these blobs again. This. And you can see our result now is only the cases where they intersect. And that's going to be the blobs essentially, right? Because we have the only, maybe I go in 2D, it'll help a little bit better. We have our blobs here. But because we did the segmented red cells to be just encompassing everything, the subtraction is going to be pretty much the same thing that if we were to create blobs. If we wanted to subtract that out, and maybe we're interested in looking on what's like the intensity of this red region on everything outside of these blue objects, we can change this to subtract. So object these red minus the blue blob. So this kind of is going to give us everything, right? So now we have this outline of everything besides where the blobs are. So if you're doing something, I know that people do like nuclear translocation assays, and this could be useful for where there you have a nucleus, you have a cytoplasm, and you want to get a measurement of the nucleus and a measurement of the cytoplasm. This is kind of where you would subtract it. And in this case, we can do the same thing. We can say, give me all the intensities of these nuclei or the volume of these nuclei here. Give me the volume of everything outside of these. And then you can get a ratio based on this. So I can say that this makes up 80% of this whole entire cell. And then these spots, these blobs make up the other 20%. And then there's also merge. This merge can be useful for when, I think, if you have maybe 2 objects that are closely situated next to each other and there's a small region of overlap, but you want to kind of maintain and make sure that they stay connected, then you can just merge them together. So this is just saying take object A, take object B, or sorry, take any of the objects within a certain class and just make them all their own entity. So in this case, it's not really going to do anything because they're not really touching. But if I were to do a merge, for example, these two objects would merge into one object. So to kind of show that, I'll go into Blender again. You can see now, I'm just doing a preview, but it's going to just, any objects that are touching that are part of the same class, it's going to combine them into one. These two are now one. These two are now. And this, I use it sometimes, but this is more of like a special case of where maybe I want to get like a full, maybe if you have like really difficult immuno and you can't really get the entire object of interest out with just a single operator, maybe you need to use two operators to get everything, then you can combine the results of those and the full volume of those are going to be merged together. Okay. And again, we can also do combining these objects into different features if you want to bin them. Something else you can also do is for these, so after you do this colocalization, if you still kind of want to get do some adjustment on these objects, you can do something called the segment morphology, where you can fill in some, like if there's any holes, for example, like that in the beginning, I kind of showed that we had this little gap here. It kind of was connecting up top, but we had this hole right in here. We can say just fill these, no, sorry, close objects. So any of these holes that are inside of it will be filled in. Same thing with if you want to erode it and make it kind of shrink a little bit. Maybe you just want to get like the border or something. We can also do some adjustments on these objects after the fact. Okay. To think with I think that covers most of colocalization. Your results will all be within here. Again, you can get the volume of this. You can create ratios based on this, and any sort of measurements that you want to pull out of this would all be contained within this objects table here.
So you have a package to calculate the correlation?
It will. It's not a inherent feature, but we can create it really quickly. So we have this option up here. Let's say we wanted to get like a percent, like a ratio. We can get this, go to feature columns here, go to create custom features. We can create one. So we'll do something like compute. We'll say we want to compute the ratio. So compute. And we would just have to figure out what it would be to. So we could take the object of one object, which would be, let's say we wanted to get like a normalized intensity, for example.
I mean the pixel-based correlations, like the Manders and the Pearsons.
Oh, I see what you mean. We do, I think we do have a built-in Pearson's coefficient. Hang on. Maybe. I think I remember seeing something like this. Yes. So we do have this down here. I don't have, if this is something you're interested in, I can look a little bit more into this. I personally don't use a lot of Pearson's correlation coefficient because there are some assumptions that are made about your data set that aren't always.
How do you get to this math?
Yep.
This math?
Yep. So we'll go over this when we go over movie making because most of these are for creating videos. But if you just right click anywhere on this region, this whole panel that we have here, there's this analysis colocalization button. And you can select this here. You can add in your two channels, pick the kind of the histogram where you want it to be, and you can get an output of this here. If this is of interest, I can look a little bit more into it, but usually when I look at colocalization, I'll look at the volume of the objects generated, because sometimes the intensities may not be the best readout of colocalization, especially if you have like more of a stain, like a higher, or if your immuno is not normalized, it can be a little bit different depending on how you imaged it. Can be a little different depending on, if you had a heavy hand when applying antibodies on day 1 versus day 2, or replicate 1, replicate 2. But this is here, if this is something, we do have the output here. You have your Pearson coefficient right here. So that's where this is contained.
That's only for the points inside the green box, right?
This is going to be adjusting the histogram, I believe. Or so the available, like if I just do this bottom, it's going to take all of these higher intensity regions. This is going to be your lowest intensity regions up here. So you can see it's getting all this background too. Here it's cutting out all that background.
Okay. Okay, thanks.
Okay, yep. Okay. Right. And that'll all be contained, all that data information will be contained within this object statement here. Does anybody have any questions? We can stop it here for today. And then because we have a lot of time left over from last week and from today and from Thursday, we can plan lie in the future, just additional days, maybe, after our meeting Thursday, we can wait a few more weeks, see what, after some members have kind of played around with it. And then if there's like one area that's not really clear, then we can kind of create a new training session based on that too.
Okay, sure.
Okay.
Thanks.
Yeah, of course. Let me go ahead and stop this recording.